Hi everyone. In this slide we'll be looking at the small intestine. Now this one is actually a composite slide, and so it contains all three different types of the small intestine. So we're going to start on this first section, and based on what I'm seeing here, I can tell you that we are looking at the duodenum of the small intestine. The lumen is right here. What we're looking at here is a villus. That's the villus at the pointer. That's a relatively flat area here, but as we move along, we're going to see a region here which kind of juts into the interior. So it's a fold that is containing both the mucosa as well as the submucosa, and goes into the interior. Okay, so in the stomach this would have been called a ruga. It would have been a transitional sort of structure, but in the small intestine it's actually a permanent structure. This submucosal fold into the interior is called a valve of Kirkring, and so this is a permanent structure. It actually serves to increase the surface area for absorption. So this valve of Kirkring is going to be there to increase the surface area by about 3 times. So there's an increase in surface area. Without this, you would have a lot less surface area, basically, about 3 times less. Okay. Now, the reason that I can tell that this is the duodenum is that right here in the submucosa, I see lots of glands. This is the submucosa. From here to here, submucosa, and I can see lots of submucosal glands. Okay. You're only going to be seeing that in the duodenum because these glands are producing a lot of mucus, which is going to be alkaline, which is going to help to neutralize stomach acids. The duodenum receives the chyme, the mashed up food from your stomach, and it's going to be coming through with an acidic pH from the stomach. And so what you need to have is some sort of protective mechanism, and that is being done right here by these glands. These are called Brunner's glands, okay, or the submucosal glands. So in these glands, you're producing a lot of alkaline mucus, which is going to be protecting the surface from the stomach acids that are coming through. Okay. Now the other thing to notice here is that this mucosa is staining very, very dark. It's very basophilic, and that's because of the abundance of lymphocytes in here. So there's a lot of lymphatic tissue in here. There's also a sign of a small nodule here. There's another nodule right here as well. Now this lymphatic tissue is necessary because what you have in the lumen is a region that is going to be in contact with the outside world, which means that if there's any pathogen that's coming through, it'll be coming in from the lumen. And so what we have here is a simple columnar epithelium, which is not very difficult to penetrate, and so you need to have some sort of protective mechanism in place just underneath this epithelium. So if we zoom in on this area, you're going to notice that there's a lot of just randomly scattered lymphocytes just underneath the epithelium. Okay, so again, that's just lymphatic tissue, just your primary point of defense. Okay. Now the other thing that is on this slide that tells me that we're looking at the duodenum is right here. This right here is the pancreas, okay, which is an organ that is just outside the duodenum. Now one thing to note here is that in this region here we have a connective tissue layer that is continuous between the duodenum, so in this region here, and the pancreas. So in this region here, the duodenum has a tunica adventitia, whereas further away we can see a free surface. And so further away what we have is the tunica serosa. Okay, so the duodenum, unlike the other parts of the small intestine, actually can have both an adventitia and a tunica serosa on the outside, whereas the other parts of the small intestine will only have a tunica serosa. Okay? Now just to get an idea of the organization, again, we have a tunica mucosa, this very intensely sort of staining region right here, tunica mucosa. So again, we have an epithelium, simple columnar epithelium. Let's zoom in and take a closer look. Okay, so you can see a nice neat row of nuclei. Maybe zoom in a little bit more. Let's go to 400x. Okay, so again, nice neat row of nuclei, simple columnar epithelium with a brush border. We can say this is kind of a dark line on the surface. So we've got microvilli there, again, very absorptive epithelium. Then beneath that we have capillaries, and we have lots of these nucleated cells. And again, all of this is lymphocytes, macrophages, eosinophils, and so on. Okay, so it's your immune system protecting you. Now beyond this, we have muscularis mucosa, which is not very prominent here, at least not in this slide, but there's a muscularis mucosa here. And then beyond that, we have a tunica submucosa with duodenal glands or Brunner's glands. And so these would open up into the bases of these crypts. Now we can see out here, this is the epithelium that covers a villus. So here, going across and down here, that's a villus. Okay? Now, when we go down here, you'll notice that it kind of splits off. The space between the villi splits off, and we get this kind of a gland here, or in this case it's called a crypt. It's called the crypt of Lieberkühn. Here's the lumen. Okay. One of the things that we tend to see in the crypts of Lieberkühn is evidence of cell division. So here we have evidence of mitosis showing up. Again, we see that here as well. Okay. What we tend to have is a lot of stem cells in these crypts of Lieberkühn, and that's because the epithelium on the surface is replenished fairly frequently. So every about 7 days or so, you have new cells being produced and replacing cells that are being lost at the surface. So cells up here at the tip of the villi tend to be removed every 7 days or so. They are lost, kind of fall away into the lumen, and they are replaced by the cells that are kind of just migrating across this epithelium, slowly moving along. And so the cells will kind of take over for them at the tip, and those cells are coming in from these crypts of Lieberkühn. And so again, if you look at this epithelium here, there is, for example, here, pretty obvious case of probably telophase by the looks of it, maybe late anaphase. Okay, so we're seeing signs of cell division going on in many of these crypts of Lieberkühn. Again, you can see that pretty clearly here as well. Okay, and so these Brunner's glands down here in the submucosa open up into the bases of these crypts of Lieberkühn, which is connected to the spaces in between the villi, and that is in contact with the lumen. Okay, so again, we have epithelium, lamina propria, muscularis mucosa, tunica submucosa with the Brunner's glands, and then we have smooth muscle, an inner circular layer of smooth muscle, and an outer longitudinal layer of smooth muscle. So these two layers of smooth muscle, that's the tunica muscularis, and then on the outside we have a tunica serosa. So on the surface we're going to have a simple squamous epithelium that covers all of this. Okay, so let's zoom back out and look at the other parts of the small intestine. Here we have another part. Now this one here is the jejunum. Again, we have one of these valves of Kirkring over here. Okay, so this mucosal and submucosal fold. We have fairly prominent villi here, and they're actually fairly elaborate as well. So one of the things we tend to see in the jejunum is that we see the kind of detached looking villi. That's just because we're looking at a three-dimensional structure in 2 dimensions. So these villi tend to branch off and be very elaborate to really increase the surface area. And so what we tend to find when we cut them in cross-section is some of them look very detached, and that's what you're seeing here. Okay, so again, these are not just free-floating things. These are actually attached somewhere. They just happen to be detached in this particular section. So these are the villi, valves of Kirkring, tunica submucosa here, tunica mucosa here, and tunica muscularis out here. And again, on the surface, we have a free surface on the outer surface. So we're going to have a tunica serosa, which is going to be mostly just a simple squamous epithelium or mesothelium with a little bit of connective tissue underneath it. Now, the way that you know that you're looking at the duodenum, sorry, not the duodenum, that you're looking at the jejunum here, is that there's nothing going on here. The tunica submucosa has no features. There's nothing in particular that will tell you that you're looking at anything specific. This whole structure is really just focused on just absorbing nutrients from the lumen. Okay, so we're just seeing a lot of very elaborate villi, and that's about it. Now let's zoom in on some of these villi just to see their overall structure. One of the things you'll notice is that the interior of these villi looks fairly empty. So here and here, what you're looking at are actually lacteals. These are lymphatic vessels that run through the middle of the villus. So this kind of looks empty. There is an epithelial cell right there. There's an epithelial cell right next to the pointer here as well. Okay, so this is not just an artifact of preparation. There's not a gap or anything. It's actually a vessel, a vascular system. So this is a lymphatic vessel. It's a lacteal. So again, things that are relatively small, they can pass across into the bloodstream fairly easily. Those will be collected by the capillaries. And so the capillaries are just underneath the epithelium here. We're going to have some capillaries down here. Okay, so for example, here, I think this region right there might contain a capillary. I think there's another one that's kind of cross-section visible right here. Okay, so that's a capillary underneath the epithelium. But things that do not pass across into that epithelium very easily, because again, this will allow for small molecules to pass across, but not larger molecules. So things like lipids will not be able to get into it. Things that are larger will be able to get into the lacteal, and they will be able to enter the vascular system by passing through the lymphatic system first. Okay, so this is a lacteal, whereas blood vessels are a little bit smaller and a little bit closer to the surface. Okay, so there's another one right here. So there's many of these things inside of each of these villi. Okay, so let's zoom back out and take a look at the last part of the small intestine. Okay, and so here we're looking at the ileum. And so again, we have our villi here. It's a villus. Now the villi in the ileum are pretty simple. They're not very elaborate. They're very much finger-like. Again, the same organization as before. So there's a lacteal in the middle of this one, for example. Okay. Here are the valves of Kirkring, so those submucosal and mucosal folds that are permanent. They're quite prominent here. But the thing that will tell you're looking at the ileum is this right here. This is a Peyer's patch. Okay. Let me see if, okay, it's been cut off here, so it's not going to be very easy to see. Okay. So right here is basically a cluster of lymphatic nodules. And the reason that it's got a specific name to it is that if I were to take this ileum and cut this, 5 centimeters further, and you get a section from 5 centimeters away from here, in either direction, I would still see lymphatic nodules in the exact same place in that section. Okay? Basically, what you have is a long chain of lymphatic nodules along the length of the ileum, and so they're not really randomly scattered anymore like they would be in the rest of the body. What you have is a fairly organized sort of structure where they're always in the same place. That's why they're given their own special name. Okay, so this is a Peyer's patch. Okay, and so it goes underneath and just goes underneath the epithelium here as well. In fact, the epithelial cells are actually also specialized. They're called M cells. And so these cells here are actually easier for pathogens to penetrate than other parts of the epithelium in the small intestine. Okay, so basically what we're doing here is we're trapping pathogens. Okay, we're allowing them to pass across so they can come directly into contact with our lymphatic system. Okay, so right there we have lots and lots of lymphocytes and macrophages and so on. So we can mount an immune response right here to any pathogens that passes across this epithelium. And again, because we make these cells much more prone to allowing pathogens to cross, the chances of a pathogen crossing here is much greater compared to anywhere else in any other part of this epithelium. Okay, and so again, you do have the lymphatic system. You have, again, the staining here is quite basophilic in the lamina propria for all of these, but you're going to have this one region here where you have these Peyer's patches where you have an increased ability of these cells to allow pathogens to cross so that they can specifically be targeted to this region. Okay. Now, the Peyer's patch is generally or most commonly found across on the opposite side from the mesentery, and so in this case here it's been cut off, so you don't really see it very clearly, unfortunately, but there is an attachment point where blood vessels come into the area. This is called the mesentery, and this is attached to other parts of the rest of the small intestine as well, and so it's just been chopped off here, but this is the entry point of the mesentery, and so wherever you have the mesentery, on the opposite side of that is where you would expect to see a Peyer's patch, which is what you're seeing here. Okay, so again, a very typical feature of the ileum of the small intestine. Okay, so I hope this has been helpful. We'll see you in the next video.